step. Okay, so the next step is known as the Krebs cycle. It is also known as, I think your book refers to it as the citric acid cycle. Okay, this all occurs in the matrix. So we're going to move that pyruvate all the way into the inside of the mitochondria. And there's a prep step. There's something that happens before we can actually start the Krebs cycle. And so there's kind of two phases to this. I think your book calls it pyruvate prep. I usually call it acetyl-CoA prep. But either way, what happens is you have this little molecule that comes in, and they represent it as a triangle in this diagram. It's really not triangle, okay? It's called coenzyme A, or sometimes it's just known as CoA, okay? And what happens is that is going to combine with the pyruvate, and it's going to create some NADHs. So we're going to give up some electrons. And also, I'm going to also make some carbon dioxide at this point. So I'm going to lose a carbon, okay? And now notice that I have a CoA enzyme with a 2-carbon molecule now. So this is what this is called acetyl-CoA. Okay? Now what's going to happen is in this thing, in the Krebs cycle, it's very similar to the Calvin cycle. I have a molecule over here that's a 4-carbon molecule called oxaloacetate, and you actually have to know that one. Oxaloacetate, and it's going to combine, and the coenzyme is going to fall off, and it's going to combine to form a 6-carbon sugar named citrate. Another one you have to know, hence the citric acid cycle. And then we're going to rearrange, and we're going to actually, at this step here, we're going to lose some electrons to NAD plus and turn it into NADH. And we're also going to we're going to lose a carbon, so we're going to make some carbon dioxide that the body will just get rid of. We're going to do that another time, lose an electron. Remember, the carbon's getting oxidized, so it's got to lose an electron, so it's losing it to NAD+. And then there's my carbon going away. Here comes coenzyme A again, not to worry. It's actually going to fall off again right here, so we can just draw it in. Okay, it comes in and attaches. There's some enzymes involved in this. And then at this step, what happens is we make a GTP. And if you remember, I told you when we did like G protein coupled receptors, they're analogous to GTP. And in fact, that will actually turn into an ATP here. Okay. And then at this step, we have a 4-carbon molecule. And now we're going to meet a new friend that's very similar to NADH. In fact, I'm even going to do them in the same color. It's called FAD, and it turns into FADH2. It's very similar. It's just a different electron carrier. Okay, we're going to rearrange again, and we're going to make one more electron carrier over here. And then the cycle will continue because remember, I had made 2 pyruvates with my glucose, and so we're going to do this cycle twice. Okay, so let's just summarize everything that kind of just happened. And really, this whole part at the beginning, we don't need to like make a big deal about, but basically what happened is in this acetyl-CoA, we had our pyruvate come in. Okay. NAD plus is going to accept that electron if we're following our electron, and we made some acetyl-CoA, just to summarize what just happened, okay? So just to summarize, per pyruvate, what went in, we had a pyruvate go in. That was it. And what came out was some CO2, an NADH, and acetyl-CoA. Okay. Per glucose, we had 2 pyruvates come in. We're just going to double everything, right? If you learn it for per pyruvate, you just double everything for per glucose. We had 2 CO2s go in, 2 NADHs come out, and 2 acetyl-CoAs. Now let's follow those acetyl-CoAs around, and let's see what happens. So we're going to summarize the whole entire Krebs cycle, okay? So we had 1 per pyruvate. We had 1 acetyl-CoA go in. We made a couple carbon dioxides. We made 3 NADHs. We made 1 FADH2, and we made 1 ATP, okay? Now, per glucose, we made 2 acetyl-CoAs. We made 4 carbon dioxides, 6 NADHs, 2 FADH2s, and 2 ATPs. Okay, sounds like a lot. Really, here's the key part. We've basically gotten rid of all of our carbon at this point, and all we have, we made a couple of ATPs that can be used, and we made a lot of electron acceptors, okay, or electron carriers. So let's see, we're going to see what happens with those electron carriers. This is like a large schematic of what's going to happen in the next step, this electron transport chain stuff. So this is my inner membrane. I'm not going to spend a ton of time on this picture because you're going to get the details on the next page. This is the matrix, and then this is the intermembrane space, the space between the inner and the outer membrane. Okay. The overall gist of this is going to be, this is going to represent my large electron transport chain, or my ETC. Okay. What we're going to do, just kind of like before, is those electron carriers are going to drop off their little electrons, okay? And the electrons are going to go through the electron transport chain, and when they do that, they're going to pump hydrogen to the other side, okay? And now we're setting up a big hydrogen concentration gradient. Ooh. And what can I do with my hydrogen concentration gradient? I can allow them to go through this molecule here, which is ATP synthase, and make ATP. This is oxidative phosphorylation, okay, using ATP synthase. If you use ATP synthase, you're doing oxidative phosphorylation. So then I can make some ATPs, okay? That's the overall thing that's going to happen. I'm going to use my electrons to move through the electron transport chain, pump hydrogens across, power ATP synthase, make some ATP, okay? Where'd those electrons come from? Notice I drew them in blue because they come from NADH and FADH too. Great. Okay. So let's get into the details. Get ready. You want to write small-ish on this picture and just kind of follow the details. Okay. Here we go. Ready? This first guy that we're going to meet is going to be named. He has the name of complex 1. So that's this one right here. The good news is they don't have really tricky names, complex 1, okay? So that's the good news. So complex 1, what happens here is NADH is going to donate its electrons, okay? And when complex 1 accepts electrons, it must also accept a hydrogen. So when complex 1 accepts electrons, must also accept an H+, okay? And that H+ is going to get pumped across. So let's just watch that happen. So here comes, and I sort of wrote in the wrong space, my NADH, and it's going to drop off my little electron here. There's my little electron, okay? And my hydrogen ion is going to get pumped across. There it goes. Okay. So now, so that's kind of what's happening.